Everybody, welcome to the Emerging Research Showcase on Genomes and Phenomes. My name is Steve Lommel. I am the Experiment Station Director and the Associate Dean for Research in the College of Ag and Life Sciences at NC State University, and I'm just going to give you a little bit of a welcome here. I want to thank NCBC for hosting us, and welcome to NC State University event. And the Emerging Research Showcase focused on genomes to phenomes. The Emerging Research Showcase events were developed as a way to introduce our external stakeholders to new research projects that could benefit from collaboration. Today, we are focused on research in the college that impacts our understanding of how plant and animal genomes are expressed in or modified for different production systems. We will hear from academic industry teams that are bridging the gap between genetics and consumer-friendly traits that impact sustainability and the important role that academic innovation and public-private partnerships play in building capabilities in the workforce. While Emerging Research Showcases typically spotlight faculty, we're doing a little bit differently this time. We are spotlighting projects that already have an industry partner. So you can all see for yourselves the value that academic collaboration provides to an industry partner. I hope you all see yourself and your institution as a future NC State collaborator. Deborah Thompson and Dinah Schuster will be your guides in that today. We look forward to identifying projects that we can implement together to solve the grand challenges that face agriculture and life sciences, and our ability to positively impact sustainability and to bring solutions out of academic laboratories and into the hands of consumers. Please be thinking about a moonshot project. If money wasn't an issue, and based on what you hear today, what project would you propose right now? So at that, I'll go ahead and turn it over and get this thing going, and thank you very much.
Thank you, Dean Lommel. I'm Lauren Anderson. I'm going to be moderating today. I'm a PhD candidate in animal and poultry science and microbiology. And the first person I need to introduce today is Dr. Christian Tobias, who's the National Program Leader for the USDA NIFA's Institute of Food Production and Sustainability. So Dr. Tobias oversees key programs like the Potato Breeding Research, the Agriculture and Food Research Initiative, and especially relevant to today's program, the Agricultural Genomes to Phenomes Initiative, which funds animal and plant science projects nationwide. He'll give an overview of this federal initiative, so hopefully folks in the room can identify alignment with the university industry partnerships featured today and our collective mission to advance sustainability in production. If we can get Dr. Tobias pulled up. Here we go, we're ready for you, Dr. Tobias.
Hi, everybody. Can you hear me?
Yes.
Okay, I'm gonna share my screen. So I want to thank Deborah for inviting me today, and I hope that everybody is well there and dealing with the aftermath. And I'm not talking about the election, I'm talking about Hurricane Helene here. I know that a lot of people there, especially in western parts of the state, have experiencing devastating losses, and a lot of infrastructure was washed away. And, in addition to FEMA, the USDA's risk management agency is there. And even NIFA has a small program that may be of interest to people there on rapid response to extreme weather events. So I just wanted to bring it up. This program accepts applications year-round on research and extension, helping agriculture become more resilient to these extreme weather events. So hopefully most of you are familiar with NIFA in the room. It is the extramural science funding agency within USDA's research, education, and economics mission area. And it invests in and supports research that ensures the long-term viability of agriculture and ensures that groundbreaking discoveries in agricultural related sciences and technologies reach people that can put them into practice. Let me go to the next slide. There we go. So some of the topics that are covered by NIFA programs include advanced technology, education, food science, food safety and nutrition. There are international partnership opportunities. We cover plants as well as rural communities and natural resources. So the Agricultural Genomes to Phenomes initiative was begun authorized in the 2018 Farm Bill. I joined NIFA in 2021, and it is one of the few programs at NIFA that involves both plants and animals. And I know that many of you in the audience may be already familiar with the program. The program team consists of myself as well as Angelica Van Gore, who is in the Animal Systems Division and represents expertise on that side of NIFA. And then our program specialist is Dan McCoy. So the purpose of AG2PI, which was in the Farm Bill, was the Farm Bill language was to just study agriculturally significant crops and animals in production environments, and to ensure that gaps in existing knowledge in plant and animal genetics and phenomics are filled, and to build a functional understanding of the relevant genes from plants and animals that are important to agriculture. Then we hope to ensure the future of genetic improvement of crops and animals, and study the relevance of diverse germplasm, such as crop wild relatives that are an important source of genetic variation that might be relevant to resiliency and provide tolerance to issues like drought that we need to address. Then also we hope to advance the genetics to reduce the economic impact of pathogens on crops and animals, and then disseminate the findings, of course, in the appropriate way. Last year, so this is a non-AFRI program, and the RFAs typically come out early in the year. And so the RFA for this year has not been released yet. But the priorities are from last year. They include promoting effective collaborations across disciplines, developing models to connect traits such as yield and feed conversion efficiency and production efficiency, and nutritional quality with environmental variabilities and genetics, to advance the data science and employ common data architectures across crop and animal systems in ways that are smart, and using fair data principles. There's also this component of phenomics and engineering novel hardware or information systems to acquire and interpret and analyze the large data sets of things like hyperspectral imagery or satellite data. Then the priority was also to study the relevance of diverse germplasm as sources of unique genes. and to improve the quality and availability of crop and animal genetic resources that can reduce economic impacts of climate change. So in 2018, the Farm Bill authorized $40 million for this program. However, we've never had that large a budget. It's never been larger than $2.5 million. So it's actually rather a small program. In 2020, we had, with a budget of $960,000, we had 4 applicants and funded a consortium of groups led by Iowa State that created the AG2PI project, which I'll go into, but really sought to build a community of researchers around this area. And many of you may have been exposed to some of their products. Then the subsequent awards in 2021 and 2022 were made to the same consortia to give out seed grants, as well as workshop grants to develop small projects. And so those about $2.5 million went out to over 23 different states and funded a lot of really interesting projects in both crops and animal systems, or both. Back in 2022, then we saw our budget increase, and we had released a competitive RFA. This RFA was for integrated research and education projects. And we awarded 2 projects, one to UC Davis and then another to Iowa State. Last year, policy made a determination that this was a research program. And so we requested research-only projects and funded 2 out of a total of 22 applicants. And the funds were awarded to UC Riverside, UC Davis, and then a conference award to Iowa State. So I'll just now turn to some of the impacts of the program so far. The AG2PI consortium itself had put on a number of workshops. Their themes were around prediction and breeding, AI and computer vision, convergence of plant and animal systems, especially around data science, democratizing access to AI and other predictive algorithms, creating a more inclusive community and building those professional development opportunities into the program itself. So they had these field days, conferences, workshops, surveys. I think the field days were really a big success, as well as giving the opportunity for the seed grant awardees to present some of their data. And in addition to the Nebraska, Arizona, Iowa State, and Idaho, there were, of course, coordination among other entities involving, like such as multi-state groups, the National Association of Plant Breeders, the FANG, which is an annotation project for animal genomes, and big data projects. So it's still the website is still available, and the content on YouTube will never go away. Looking the other day, there were about 29,000 views on their YouTube videos, and there are over 98 of them. So I'd encourage you to look at those and check out some of that content. This model was not without challenges, of course. The most commonly reported challenges were just had to do with managing so many seed grant awardees, difficulty with their institutions getting the award monies into accounts and hiring, and delays in completing the deliverable that resulted from directly from these challenges. So that was one of the reasons that factored into us moving away from this sort of model to competitive research model. One of the first awards then that was made was to a project involving members of the original AG2PI team, but also several other animal scientists. And the goal, which is really an interesting one, is to promote public-private partnerships by developing new technologies for data encryption to encourage private partners' participation in some of these research efforts. Because this particular challenge is important especially with animals, because no one institution has the capacity to have large herds of animals that they can collect data on. So the animal research community, of course, relies on the private sector for that. So this idea of data encryption, but still allowing the analysis to be done and the results to be valid is really exciting, and hopefully will spawn more collaboration. I know that this was work that started as an AG2PI seed grant. The PI is Hao Cheng at UC Davis, and then it went on to become a full-fledged research project. The other award in that first competitive year of AG2PI went to Iowa State to look at, and Nebraska, to develop genome-wide association mapping studies and data sets, as well as transcription-wide association and EQTL mapping on corn, sorghum, soybean, and swine. And then one of their objectives was to develop the open source R packages and Python libraries, as well as to interweave the research and education components through the training in cross-disciplinary research to produce the skilled STEM agricultural workforce. So in this case, these two projects were the integrated projects. And then it was recently announced that the awardees for the 2024 fiscal year that included UC Riverside. This was a study that is being conducted on heat resilience in U.S. honeybee stocks, on the genetic basis for that. Then another award went to UC Davis on strawberry breeding for nutritional quality and flavor. And then an award went to Iowa State's Chris Tuggle for putting on a workshop in conjunction with the AGPT Ag this summer on single cell genomics. So these are the projects that the initiative has funded so far. Just to add some additional information that might be of use to the audience here, there are upcoming NIFA opportunities. So I mentioned the AG2PI RFA. That's coming out in mid-January. Some other upcoming funding opportunities that may be of interest to this audience include some in the area of biotech risk assessment, or BRAG, methyl bromide transitions, the Organic Agriculture Research and Extension Initiative, and then the new AFRI, Foundation and Applied Science RFA, will be likely coming out in next month. There are also educational programs such as Higher Education Challenge Grants programs for helping to build curricula. Then the other programs they're listed that are coming out in January, relevant to plant and animal systems. So I'd encourage you to take a look at those, and also to sign up for NIFA updates to have those opportunities sent to your inbox directly. I think I'll wrap up by just directing you toward some of our resources at NIFA for funding opportunities, how we run competitive grants, how to prepare competitive grants, our policies on everything is in the policy guide, and then a short site on describing our indirect cost rates, and how to use the grants.gov, through which all of our incoming grants are received. So I thank you, everybody, for your attention, and I would be happy to answer any questions that you have right now.
Do you have any questions for Dr. Tobias? Go ahead.
Hey, Christian. Hi, Christian. This is John Salmon. Nice to see you.
John.
All of the awardees I saw for AG2P were university. Are companies eligible to apply?
Yes, especially as consortia, but they are eligible entities on their own. The germplasm sharing, I should mention, because USDA recently came out with a press release on germplasm sharing, and a lot of people, it caused some angst among people, but the policy has not really changed at all for NIFA. We encourage all forms of intellectual property to be pursued, but also we do require you adhere with our departmental memos on data sharing. So the data sets from federally funded research do need to be publicly available.
I have Poultry Science's own Dr. Allison Pullen, and she will be joined by Dr. Brooke Bartz from Blue Buffalo R&D at General Mills, who's going to be joining us virtually today to talk about broiler production, the relationship between the genetic strain and production environment.
All right, well, thank you so much for the introduction. So yeah, I'm Allison Pullen. I'm from Poultry Science, and then my colleague Brooke Bartz from General Mills is online and will be co-presenting today. And so the main challenge that our team was interested in tackling is in broiler production. And so for those of you without a poultry background, broilers are chickens that are raised for meat production. And pictured here on the slide is what we would see in a very typical broiler production environment here in the US. So we have birds that are raised in an indoor environment with automatic ventilation. They have ad libitum access to feed and water, as well as bedding on the ground to help absorb excreta from the birds, and rigorous biosecurity protocols to maintain the health of the birds. In these environments, we also use fast-growing genetic strains of broilers so that they can reach their target weight for processing in about 6 weeks. So while this is a really efficient and safe way of poultry production, there have been valid animal welfare concerns that have arisen from conventional poultry production, specifically where we use fast-growing strains. As the birds become larger and as they age, they're more susceptible to leg and foot health issues that actually impair the bird's walking ability. So there's already been ongoing discussions within the industry about how to improve this, but these animal welfare concerns have also come up with the general public, retailers and restaurants that purchase chicken meat, as well as animal activists that have highlighted these welfare issues on their websites and with undercover videos on YouTube. And so with these animal welfare concerns, it does create a social sustainability risk of the acceptability of these production practices. And so there's really a need to continually to improve animal welfare and broiler production and think about what types of evidence-based standards we're using for animal welfare. So one example of these animal welfare standards is the Better Chicken Commitment. And so this is an initiative that has a variety of different animal welfare standards for the indoor environment. But one of the main tenets of the Better Chicken Commitment is to actually source chicken meat only from farms that are using slower growing genetic strains. So these slower growing strains are going to be reaching a target weight anywhere from 8 to 12 weeks of age compared to that conventional strain that's hitting it at 6 weeks of age. However, most of these slow growing strains, there's a list that they have on their website. Most of these strains are actually not available in the US. They're primarily hatched and produced in the UK and in the EU. And so the commercial viability of using these strains in the US is quite limited, even though we have over 200 restaurants and retail groups that have actually pledged to the Better Chicken Commitment in the US to source chicken meat from these farms. Another area of animal welfare that has been a discussion in the industry is, again, thinking about that environment piece. So in conventional environments, environmental enrichment, which are structures that provide additional behavioral opportunities for the birds and may be able to also benefit leg health for birds, are rarely provided. So thinking about environmental enrichments as well as outdoor access production systems. Outdoor access allows birds to leave the indoor housing and have access to more space outside and perform species-specific behaviors such as foraging on the range. So this is also an area that companies are looking into as a strategy to improve broiler welfare, because it's presumed that having this extra access would improve their welfare. But when we look at actual research in outdoor access systems, the welfare outcomes and productivity outcomes are highly variable. And one of the main reasons of this is because of a wide variety of genetic strains and ultimately the suitability of genetic strains for those types of production environments. So if we start to switch to different management strategies, whether that's with a slower growing strain or an outdoor access environment, we may create significant economic trade-offs by using a less efficient bird or getting more highly variable outcomes in a different production environment, which could actually threaten the economic sustainability of broiler production. So thinking about that social sustainability piece that I mentioned earlier and the economic sustainability piece, we want to try to find a nice balance between the two and have evidence-based guidance around animal welfare, as well as the chicken meat product, thinking about food safety and meat quality metrics, as well as economics to actually justify that switch. So now I'll switch it over to Brooke.
And this is where the partnership with General Mills comes in. So based on the challenges mentioned by Allison over the last few slides, it was quite evident that there was a need not only in the research community, but by the industry to identify animal welfare programs that not only made sense for the birds to express their natural behaviors, but that could also be economically feasible long-term. In order to achieve this, there would need to be a need for unbiased research to be compiled together to assist in the transformation of poultry rearing systems. Next slide. So, grand scheme of things, General Mills is a pretty small player in the poultry industry, in which we source less than 0.5% of the total broiler market. However, just because we are a small player in the broiler market, this does not excuse us from supporting the advancement towards higher broiler welfare. Next slide. And so the way we're able to do this and support our commitment towards better welfare is by sourcing birds that have been raised under GAP certified programs. So these programs include more space for birds, enrichment, and processed using controlled atmospheric stunning systems, and only using breeds that are certified under these programs. So I will go ahead and kick it back to Allison to chat a little bit around the collaboration work.
So our team between NC State and General Mills, in order to get this comprehensive interdisciplinary look at animal welfare and product outcomes, included me on the animal welfare side, and then Dr. Lynn Walker from Poultry Science, who's a microbiologist who focuses on food safety of poultry products, Dr. Yan Campbell from Poultry Science, who's a food scientist that focuses on the meat quality of poultry products, as well as Dr. Tom Buchina from Ag Economics, who helped us with economic modeling and economic analyses, as well as Brooke. And so our first step in really in trying to understand kind of what evidence we have for thinking about comparing genetic strains and different production environments is we wanted to do a literature review. So General Mills sponsored a project with us where we went into the literature to compare conventional versus outdoor access systems in the animal welfare, food safety, and meat quality metrics. And this was where we learned, as I mentioned in the background, that actually comparing these systems directly is quite challenging because of the high variety of genetic strains that have been used in the research. And then the majority of the research has also been conducted outside of the United States. Most of it's been conducted in the UK and in the EU, which has different climates from the US, and climate is a main factor that drives how much broilers will actually utilize that outdoor range area, and therefore how many benefits they can get from that range. And then Dr. Buchina did some economic modeling based on US production system characteristics to compare production systems, where we found that switching to an outdoor access production system would increase the break-even price for producers by about 269%. So because of this limited ability in being able to compare the production systems because of this high variety of genetic strains, we then partnered with General Mills. Let's see here. They gave us a gift so that we could then pursue a live trial where we could then actually have a more controlled way of comparing these production systems using the same genetic strains in each production system to really understand the trade-offs in all of these metrics. So we used a fast-growing strain of a Ross 708, which is the most common genetic strain that's utilized in the industry. And then we used a Sasso slow-growing strain, which is part of one of the hybrids approved by the Better Chicken Commitment. And so for the target weight, Ross would hit this in 6 weeks, and then Sasso would reach the target weight in about 8 to 9 weeks. We had a conventional environment at our NC State Chicken Education Unit, and then we partnered with a local poultry producer in Angier, North Carolina for our outdoor access environment. And this was a silvopasture environment, which has tree cover spread throughout the range. And we actually conducted this research. It just finished last month. So we ran the trial from August to October. Again, looked at a variety of performance metrics like feed conversion ratio, average daily gain, welfare metrics, including behavior and health outcomes, food safety, salmonella, microbiome, as well as meat quality metrics with carcass yield and a variety of pH, meat color, and then they're also going to do a sensory panel. So the data analysis is still underway, but by being able to have this more controlled live trial from what we'd seen in previous research, we are already seeing differences in kind of how the strains were responding to each environment, such that that fast-growing strain had more challenges in the outdoor access environment, where we saw higher rates of, or higher prevalence of salmonella, as well as a higher prevalence of severe lameness or mobility issues and foot pad lesions. So this data analysis is still underway, but we're looking forward to the results.
So the great thing about this collaboration is that it was allowable then for all parties to benefit from the data set. So we were extremely mindful in how funding was given so that the literature review and the results from the live bird study would be submitted to open access journals. It is anticipated that there will be at least 3 additional publications in addition to that first literature review mentioned by Allison, and those will be submitted once the data has been analyzed by the respective labs within the poultry department. Additionally, it was great to see how our financial gift was able to fund several graduate student stipends and develop new relationships within the poultry farming community. Next slide. So we'll anticipate the results of these studies that will allow for the inform of an entire industry and give an evidence-based approach as we continue to strive towards better welfare parameters as a whole. So with that, I'd like to thank the poultry department for their continued collaboration and open it to any questions.
And then, okay, great. And I guess we're maybe doing questions at the end. Okay, great. Awesome. Well, thank you all so much. Appreciate it.
All right, go ahead and make note of your questions and hold on to them for the end of the session. Next, we have Drs. Acer Van Wallendale from Horticultural Sciences and Dr. Emily Bellis, who is the computational biology lead at Avalo Biosciences, talking about genomics and crop diversification.
All right, so this is the problem at Avalo that we focus on solving. It's this long time frame for developing a new crop variety. So whether that's using traditional breeding approaches or even approaches using genetic modification, even though those are fast in the lab, they can take a long time to get new varieties out in the market through the development process. So at Avalo, we really focus on that gap, especially given the rapidly changing climate. That delay between development and deployment of a new variety is a really big problem. So we do that with a few tools. So one of the tools that we use are advanced computational approaches based on interpretable machine learning. So our patented, we call it the GDIP approach, so gene discovery through informationless perturbation, allows us to identify causal genes linked to phenotypes with higher precision and with a much more strongly controlled false discovery rate. So that's one of our tools. But the other one is really this ability to leverage genetic diversity in a wide variety of species. So these 4 species that I've indicated here, these are what we refer to as the formidable 4. So they are all crops that have really outsized impact on the environment, but they are also really important for the economy, and then in some cases as staple food crops as well. So for example, we have projects in rubber dandelion, which is an alternative source of rubber. That is not even a domesticated crop, but it's related to the weedy dandelion that you may be familiar with. And in that project, we're really trying to increase the latex production in these dandelions. And then we also have internal programs where we're directly using crop wild relatives, land races, and other really diverse germplasm directly within our breeding program. So I want to give you one example of a project that we've been doing in rice. And so this is a project that's in collaboration with Arhelia Lawrence at Arkansas State University, funded by the USDA. And what we're doing in this project is combining these 2 different tools that we have in our toolkit, right? So this improved methods for gene discovery along with genetic diversity, in this case, in diverse land races of rice. So rice was domesticated, one of its major centers of domestication in the Yangtze Valley. But after domestication, it spread and diversified into many water-limited environments, like rain-fed lowland environments. So what you're looking at here, this is 1,300 rice land race accessions. So that's each of the points on this figure. And it's overlaid upon a map of habitat suitability for growth in rain-fed lowland environments. So we do genotype associations with that underlying habitat suitability map to identify candidate alleles associated with growth in water-limited irrigation systems. And so that project is currently underway. We're following up on many of the candidate alleles that we identified first. We're doing high throughput phenotyping, and that part of the project is led by our Hilia's lab and a postdoc graduate student in her lab. But what I really want to emphasize is a lot of this project is really driven by knowledge of the evolutionary biology of the species, right? So that's, I think, part of the reason why we're really enthusiastic and interested in partnerships that allow us to use knowledge of crop domestication history and evolutionary biology in our development of new varieties.
Thanks, Em. Right. So that's where I come in. The main question that I'm really interested in my lab is what can we learn from weeds? So one way to think about this is that in the evolutionary history of every crop, there was a progenitor, and someone decided it looked interesting to eat, and they domesticated it over time. And in many cases, the sort of ancestral population that it was domesticated from died out, but in a lot of cases, that ancestral population did not die out. It may exist in a natural system, or it may exist as an agricultural weed. The weeds that survived as sort of offshoots of the domestication process, I think are very interesting because they often exist in very similar landscapes to where the crop is. Sometimes they can actually reinvade the cropping system and they can cross with it, form all kinds of messy genetics. But they often have totally different genetic histories. They have different selective pressures on them. And that results in a lot of different potential for varieties of genes that may be beneficial to crop improvement, especially in a changing environment where we need crop diversification. So the weed that I have spent the most time with is this one that most of the folks in this part of the world haven't heard as much about, but it's more common in both in Asia and in sort of the dry region of Colorado and Nebraska. It's called proso millet. It was domesticated very early on, around the same time as rice in China around 10,000 years ago. It has since spread throughout the world, and it's mostly in the US used as bird seed, but in other parts of the world, it is a really high protein grain crop. And there's even some sticky varieties, so you can use it like sticky rice to make mochi. I'm interested in it as an invasive weed. So in many parts of the world, along with domesticated proso millet, there is a variety species. People are in disagreement about what it actually is. That looks totally different, is considered very closely related, but occurs in similar locations to the domesticated crop. What I was able to do was go through North American populations of wild proso millet, collect samples from around the US, and also get some samples from the native range in China, and compare all of those weedy proso millets to different populations of domesticated proso millet. And Emily talked about really complicated statistical models. I used a very uncomplicated statistical model here, which scans across the genome and looks for differences, basically subtracts one from the other. So we get this marker called, or this statistic called FST. And what you need to know about that is when FST is higher, the genomes are more different from each other. So this allows us to identify parts of the genome where weedy millet is totally different from domesticated millet. And these, sometimes they just look like noise, but oftentimes they line up with genes of interest. This is my favorite one at the moment here. It's called GN1A in rice. And it's a locus that controls grain number by influencing the accumulation of hormones within the flower bud. So we see that this grain number locus is differentiated between the populations of proso millet. What can we actually do with those data? So one of the potential downstream applications is as a precursor to the sorts of things that breeders like Avalo do, we can take these different versions of the gene for this grain number locus, and we can use genetic transformation to introduce them to different lines and see if they actually produce the desired effect of increasing the grain number. And this is not necessarily to release these varieties into the wild or into the into cropping systems, but to highlight the importance of potential loci that breeders can then select on in the future. So we have a lot of different traits that we've been able to find differentiation for, from grain quality. A weird one that I accidentally found out about because I tried to plant these in the field and forgot that they're bird seed, is bird resistance, where the weedy varieties will actually not get eaten by the birds. And also that stickiness trait that I mentioned earlier. So with that, I wanted to wrap up and say that, and I hope Emily will agree with me, that accelerating breeding for modern agriculture requires these advanced data analysis methods where we're able to take whole genomes and assess new traits and rapidly introduce them into breeding lines. We're both interested in crop-related weeds, from the dandelion to the proso millet, as a rich source for genetic material and potentially stress tolerance for weedy or for domesticated plants. You want to say something else?
No, that
again?
was great.
All right. Thanks very much. I hope to talk to folks later.
There you go. Thank you so much for having me. And I'm an animal geneticist and a quantitative geneticist. So today I'm going to give you a little bit of a sort of something that has been for a while in my radar, but it's also a little bit out of my comfort zone. I don't have a particular partner here today, but I will try to highlight the partners I've been working with throughout the last 4 or 5 years to work on this area. Just as usual, this is not just the work of myself, it's the work of my group. So those are the ones that truly do the work, and I'm just here to spread the gospel. So kudos goes to them. I normally put them in front because then I don't forget to do it. So this is the people that do pay, and as you can see at the bottom, since those are the 2 main partners that we have been collaborating with. Clearly Smithfield, for anyone who works in the swine industry, and North Carolina, a large partner for us, but also the Mashup and Acuity now, Mashup before Acuity now. Can't keep track with the change of names, but big collaborators. They have been collaborators before then. They are collaborators in this. We hope that they will be collaborators in the future too. So if I don't mention that the work has been done mostly with these 2, so you don't see the logo at the bottom, but it has been done with them. So what I'm trying to very quickly convey is that I'm a geneticist, so I'm interested in the potential of the microbiome as both a mediator of environmental variability and physiological status of individuals, but also as a source of potential selection. So the things that we have been working on in the last few years have been trying to understand what the effect of the microbiome was on efficiency and resilience for swine. Evidence that the composition of microbiome as a genetic component, and if this was 5 or 6 years ago, probably I wouldn't have a definite answer, but right now it's yes, there is a relatively large genetic component on the microbial composition, but also some of the evidence that microbiome also serves as this proxy for the physiological status of an individual as a tracker of the environmental variation. So it serves as an intermediate omics. I will skip, obviously, all the nightmare complication that arise from the fact that microbiome sits exactly in between all these things altogether. But what we're interested in are mostly related to efficiency of production and lately more on resilience. And why not? Swimming pigs are always good. So I already mentioned this, so I will skip this, but I like the microbiome because it sits between what I can measure and what I can predict with, okay? So that's why I'm interested in. So I'm going to provide you snippets of research that has been done over the last 4 or 5 years. I apologize, I will skip 99% of the details. If you're interested, I'll probably have papers or you can reach out to me. So the first thing we wanted to see is like, we know we can select individuals for increased efficiency. There are some problems in doing that. We know that the microbiome has a component related to it. Can we reintegrate the microbiome as a source of variability? If you think about it in terms of environmental variation, or a source of selection, if you think about selecting. So this has been done in collaboration with Acuity. See, I put it there. So the idea here, we had some sort of master design where we actually collected microbiome and genomic information in different populations, and we wanted to characterize whether the microbiome was associated with a particular composition of microbiome was associated with growth parameter, if there was a core component, and if the host had a particular effect. So a lot of information, and this thing tend to be packed with graphs that convey little information piece by piece. But on the right side, you can see that the yellow part is the genetic part of microbial composition, and that the bar stacks to how much it explains of the total variability. And so for microbial variability, so there is a genetic component, and we were able to identify some of those core taxa that were significant for it. So a few other things. We already know that there is genetic variability. We wanted to track whether that's genetic variability. This was done with Smithville, whether that genetic variability was translating into difference in microbiome. And the short answer is yes. Clearly, paternal lines that have been mostly selected for grow efficiency have a diverse microbiome than maternal lines that have been selected for reproductive efficiency. And we have seen this both when we measure feed efficiency per se, but also more interestingly, considering that microbiome is related to the gut-brain axis, also to traits that are related to the behavior of the feeding behavior and the amount and the time spent feeding. Again, I will not go into details, but you can identify taxons that are differentially expressed in efficient versus non-efficient, and also more interestingly for me, that are differentially expressed in paternally selected or maternally selected breed. Same thing here. Clearly, because I'm a geneticist, I do want to see if there is a genetic component to all of these things. There is a genetic component to all of these things. You can estimate microbial heritability, which is what you see in the bars here. And if you're truly ambitious, you can identify some genes that is actually related to microbial abundance in the pigs. We're doing still some work into this, but again, this has been done with Acuity. I keep going back and forth. And if you're really ambitious, you can try to establish causality, whether the effect of the microbiome, whether the genetic effect is mediated through the microbiome or it is directed through the microbiome. And because we are in the business of predicting things, you can also use the microbial information to predict phenotype, just as any other intermediate omics. And we've shown it. I'm not the first one. I'm not going to be the last one to show it, but we'll actually be able to predict across population, so that we can use microbial information collected in one population to predict phenotypic information, phenotypic performance in another population. So the idea here is that we can close that G by E gap, but measuring something that we cannot capture in commercial environments, but we can capture through microbial information. Then we can switch a little bit and think about, this is done with Smithville. You can switch a little bit and think about it in terms of resilience. Resilience measure as the deviation from the expected feed consumption. There's been a lot of research that we've been doing, us and many others, about measuring efficient resilience of an individual as the potential of not being perturbed when the environmental challenge is going on. So if you have environmental changes, either that they're due by changes in temperature or some viral infection or bacterial infection, you can measure whether the animal was expected to eat a certain amount. If there is a dip, you can use that to measure whether the animal is resilient or not by measuring how much the dip versus what you would expect the animal to consume. And clearly you can reinsert the same sort of exercise we've done before. Are there genetic differences, and are these genetic differences sort of mediated by the microbiome? And the answer is yes. In this case, we're trying to categorize whether the low resilient and high resilient individuals are different. The gray one here are the low resilient, and the other colored one are the high resilience individuals. And clearly they do have a genetic, they do have a different microbiome. Sorry, I went forward. And we can, if you're actually really ambitious, you can try and do some prediction and try to say, if I measure the microbiome in an individual, can I predict whether it will be resilient or not in face of environmental challenges? And the answer, always with a million of caveats, is yes. This will lead to a little bit of the last piece of information, which is working with this company for me, for us, has been extremely rewarding, not only because it provided access to data, provided access for them to our research, but also allowed us to leverage very effectively grants money from USDA in this case. So related to all this work, there are 4 AFRI grants that are currently either finished or in the making that are looking at heat stress resilience and microbiome and genomic information. This one is improving through genomics or using a series of intermediate omics. And I'll skip all these because we do not have time. It's going on right now. But we have some interesting and exciting results to share, I hope, next time. And there is another one that has been related to the same kind of work, but related to in utero heat stress. So taking the same exercise, not only from heat resilience or heat tolerant or not heat resilient sows, but also to the next cycle, and see how in utero heat stress determines the performance of the next generation. But to a certain degree, there is actually something else. There is reusable data that comes out of all these projects that come from different aspects. So we were able to get a pangenome project funded, which again involves both Smithfield and in this case PIC and DNA genetics, which kind of reuse some of this information that we have generated from the previous grants. So I throw a lot of things to you. That's what I tend to do. But if you do have any question, if there is anything that wasn't clear, of course there was anything that wasn't clear, you feel free to ask question at the end or stop me anytime you want. Thank you.
Thanks, Dr. Malteca. Up next, we have Dr. Jeff Dunn in Crop and Soil Sciences, who will talk to us about improving roast peanut flavor using machine learning approaches.
Good afternoon, everyone. So I'm Jeff Dunn. I'm a peanut breeder at NC State. So today I'm going to talk, and I apologize in advance, we got a little too ambitious with the number of slides. So if you're used to drinking from the fire hydrant, here we are. So today I'm going to talk about more of a story as to how we got to this project, the kickstart of this project. So we actually haven't even started this. This is start project of next year. But basically, I kind of wanted to give you an overview of all the work that's gone into essentially, developing our partnerships with some of our industry players that are key in terms of flavor improvement. So the first kind of introduction here is actually the partners that we've been working with on this. So I'm a part of the NCSU Peanut Breeding and Genetics Program. And then my industry partners, unfortunately, could not all be here, but I'm going to introduce them as the companies that we're working directly with. So Marge Wrigley, everybody knows them as our candy, confectionery, snack food company extraordinaire. Qualisense is a seed quality kind of engineering, image-based, type software company. And then JLA, or John Leake and Associates, which is more of like an analytical chemistry or service kind of lab. But they also provide for the peanut industry a flavor descriptive sensory panel for us to make flavor selections on. And so what is actually the problem when it comes to breeding? So we kind of heard a little bit about the kind of stagnation of a breeding program, how long it takes, but then all of these things that affect, the diversity as we're making selections toward eventual releases. And so as we're going through, we make initial crosses, we go through the, kind of the easier selections, our qualitative selections, and then we get more into the more quantitative kind of selection approaches, things like yield, drought stress, things that are are like kind of hard to quantify. And then at the very bottom, you see we get into this, kind of national program testing network, which is called the Uniform Peanut Performance Test. And if you notice at the very bottom, I know it's very small, but we're actually only submitting 2 lines. So we get 2 lines of data that's taken across, or 2 lines that are submitted across multiple environments, and we're getting flavor panel assessment on them. But if anybody knows anything about a genetic diversity of 2, it's essentially nothing, right? So we can't actually make selections to improve flavor. So there's where our problem comes in. So how do we actually do this? So from a descriptive sensory panel, we actually make the selections based off of subjective evaluations of a lexicon of different flavor topics, right? So we have things like roast peanut flavor, sweet aromatic, dark roasted. All of these things go into there, and they categorize all of these things and make like an evaluation on our lines. So the first one was actually developed here at NC State in the 1970s. Some of the categories in that lexicon have changed over time, to include things like fruity fermented, so things like off-type flavors. And basically what we ended up doing was we started talking to kind of our consumer products groups like Mars Wrigley, and they asked the very simple question, is flavor declining in our U.S. breeding programs? And so we actually put out a study in which we reviewed all of that UPPT data over the 17 years. And specifically within our program, we noticed that roast peanut, which is actually the main score of what everybody attributes to peanut flavor, is actually declining in our program, which is not very great to see. Other categories like sweetness, sweet aromatic, those are improving. We're reducing cardboardy, kind of off-type flavors, fruity fermented flavors, but we really want that robustness of that roast peanut flavor score. So what do we do from a breeding program standpoint? Well, we start scouring for germplasm that actually has an improved roast peanut flavor score. So one of the cool articles that I kind of came across is this NPR article. In it cites my predecessor from the NC peanut breeding program, Tom Islib, in which he was working with a number of groups to identify this Carolina African runner. And apparently this has kind of the most intense flavor profile of any peanut that any chef has ever worked with. And so the chef in South Carolina was very interested in finding this. We just so happen to have it in our germplasm seed stock. It was labeled Carolina runner number 4. So we provided that seed to a grower in South Carolina who now is growing it full-time for this chef in the Charleston area. And so what we started thinking about is like, well, we have 740 lines. Let's just kind of randomly select some of these and see if any of them are very good at flavor. So we ended up with 210 lines that were randomly selected based on seed quality and availability out of 740 that we have in our gene bank. We kind of did some flavor testing with a sensory or descriptive sensory panel. We grouped these based off of things we thought were good in terms of flavor, relative to that lexicon, some things that were kind of fair, had good scores, but were like really poor in some other areas, and then really, really poor. And it turns out that we actually clustered a lot of so the lines that you're seeing up there are actually our current varieties, and they are clustering very heavily with the poor scores, which is, kind of what we found in our kind of evaluation. What we ended up doing was we found these 2 lines, so this Chimera and NC Bunch. And the reason we selected those is because we represent the Virginia market type. I always tell people, we're the baseball peanuts. So if you ever go to a baseball game and get a roasted peanut, it's probably coming from our program. We supply about 95% of Major League Baseball, which is kind of a fun stat. But that being said, we want to also select things that represent our program. And so this Chimera and NC Bunch were 2 lines that we wanted to select. And then we crossed those and made populations based off of one of our kind of flagship cultivars, Bailey 2. So remember that we have these selected categories, good, fair, poor, and then we made these 2 populations with Bailey 2 in an effort to try and select for better flavor. So that being said, we have a number of lines now, right? So we have 300 plus in each of these populations. How do we go about screening these things? Like, we can't just submit them to a flavor panel. They only do about 2 or 3 at a time per day. It costs about $250 a sample. And then on top of that, in terms of our selection efficiency, we don't really know what to do with that data yet. So are there better screening methods for us to at least kind of whittle down those numbers? So we kind of scoured the internet, all the different publications that are available. And we really found only one that was non-destructive and was based on raw peanuts. And that was this HSVC, this headspace volatile concentration. And really all it was doing was just detecting ethanols. And so those ethanols are directly contributing to that fruity fermented off-type flavor, but really nothing else that is impacted in terms of the actual roast peanut score. So at the time, we'll kind of depart from like what the flavor part of it is. So at the time, we're kind of working with another company as a part of our breeding program called QualiSense. And what they do is they provide a machine or a type of equipment that allows us to do, quickly and efficiently seed sort. So when we want to select on things like larger kernel size or different shapes or colors or any of those kinds of things, and most importantly, NIR. Also at the same time, Mars, Wrigley, QualiSense, and GLA are also talking in conversation about flavor. So this is kind of a side conversation that I was not privy to. So what is a Q sorter? So basically this video kind of just depicts what a seed sorting mechanism is doing. It's sorting seed about 30 seeds a second, where we couldn't do that physically. And then we can build models to kind of predict our distributions on seed size relative to our genomic backgrounds. But what's most important to us as a variety development program is releasing based on seed purity. And in this case, we release high oleic peanuts. High oleic, the trait high oleic is basically increasing shelf life and also reducing rancidity when it comes to our products when they go to market. So this machine, from an NIR standpoint, allows us to separate those or sort those kernels based on high or low oleic values. So simultaneously, Mars, QualiSense, and JLA are working on different attributes to apply to this Q Sorter Explorer. So they're looking at different protein contents, oil contents. From a flavor standpoint, they're looking at sugar content, sucrose, different tocopherols, and then they want a regression, or they want to model using regression to implement these within the Q Sorter Explorer. But my thought was, well, why can't we take this one step further? We get full spectrum readouts on all these kernels. Why can't we just apply either some machine learning or deep learning models to predict flavor panel outcomes? And one of the success stories on that is actually in coffee. And it's relevant because QualiSense also works in coffee. They actually kind of cued me into a lot of these things. So what is in this paper is basically highlighting that they take these NIR spectra, they predict out all of these different categorical roast coffee scores, either floral, fruity, roasted spices, all of these things that are all a part of our lexicon. So why not, apply the same kind of concepts to an equally complex trait. So they developed some models, fairly accurate. What is the most interesting part of it is not only could they detect single flavor categories, but also multiple flavor categories. So if a particular roast was providing a floral and or a spicy note, that they could actually detect these, and then they can break those down based on wavelengths of known, different analytical chemistry backgrounds. So that brings us to our project. So our project is improving roast peanut flavor using machine learning approaches and applied peanut breeding. And so we got finally this summer, we got all three of these big groups in the peanut industry to come together to kind of fund a project. So how this project overview kind of looks, we already have a Q sorter. We needed something handheld because if you remember when it comes to like the flavor panel, they're doing roast paste samples. So we have to have something to be able to measure the paste. Since the Q sorter is only doing kernels. So the way that this method kind of is applied, we have we have the Q sorter. It evaluates the raw sample, gives us some spectra. We have the handheld sensor, evaluates the roasted pasted sample, gives us some spectra. And then we have JLA, who does all of the flavor panel testing for us to give us our classifications. And so basically what we're going to do is build some machine learning models on an initial base population. And then we're going to apply those models to our Bailey 2 by Chimera and NC bunch populations. And what that'll effectively do is allow us to now select within the confines of our actual testing program, as opposed to waiting to the very end, where we still have some genetic diversity left in order to improve the trait. So that's all I have. And I think we're taking questions after. My contact information's up there, so if you don't get a question out there.
Well, while he's loading that, so about 10 years ago, collaboration between the North Carolina Department of Agriculture and Computer Sciences and NC State formed what we call the NC Beef System. And what that did was we have 8 beef research stations across the state from the coast all the way out to the mountains, and we unified the systems of how they're managed in order to create a larger population of Angus-based cattle that more reflect our industry needs. And since that time, we've continued to grow and develop our faculty team. Listed here on the slide is 4 of our key main faculty that are involved in this NC Beef System. And it is an interdisciplinary team from nutritionists to physiologists to animal breeders to the animal health component. And so we've been able to expand on what we're doing based on this camaraderie we have within our team. And we've recently expanded out since hiring Dr. Weaver to include the sheep industry in this. So now we're focused on a general ruminant approach to addressing some of these industry issues. So for years, this is not the right slide set, but it's all right. For years, the industries have developed what we refer to as expected breeding difference or expected progeny differences, or EPDs. In the sheep side, it's estimated breeding values. And these traits are methods that we have in order to educate our producers on selection criteria for various output traits. And you can see this is just a simple example. I'm not promoting these sires by any means, but to give you an example, we're looking at growth traits, or we're looking at maternal traits or carcass traits, again, looking at that end product. But as we continue to have challenges in reduction of acreage devoted to animal agriculture, as we see some of the challenges involved with climate change and the greenhouse gas emissions, some of the areas of our industry have changed to identifying animals that are going to be more feed efficient. And one of the ways that we're really addressing feed efficiency is being able to identify animals that have a higher residual feed intake. And simply describing this is through one of the graphs from our collaborating partner with Vitelli. And simply put, we have 2 animals here that have both the same average daily gain here in the blue bar, but we can see in the orange bar that one of these animals is superior as far as feed intake because they are consuming less feed in order to have that same average daily gain. And so this is going to become a very desirable animal because 70% of our production costs in the ruminant industries are coming from our feed cost. And so if we can reduce some of that expense to our producers, we can accelerate that opportunity. Again, when we look at our feed efficiency side of it, the lower or the more negative number, the better that animal is going to be. So a couple different ways we can look at this is the number of pounds of feed per gain of animal, or again, that residual feed intake as a methodology to enhance this level. In addition to having some of these production challenges in the Southeast, we have some phenomenal environmental challenges on our ruminant production system as well. We know that we need to continue to increase the production of animal-based proteins, and we know that in order to meet the demands of the consumers by 2050, we need to increase the growth of the beef industry by 70%. That growth has to come in semi-tropical and tropical environments because that's where the land mass is available in order to grow those individuals. And so now we're going to take individuals that are generated in the Midwest or out in the Pacific Northwest, where our genetics are coming from, and we're exposing them to the hot, humid environments of North Carolina. And as you might imagine, they don't necessarily grow very efficient at that point. So we have these increased heat stressors. And then also in the Southeast, we have some phenomenal forages, but these phenomenal forages also have a bad reputation as they contain a endophytic fungus that creates another problem. That other problem is known as fescue toxicosis. And so what happens is when we combine that heat with this endophytic fungus, or the output from the endophytic fungus, which is an ergot alkaloid, we just have animals that are very poor doing, as you can see in that far picture there. these animals then are not going to be that feed efficient ideal model. So then the goal of this team is to devise a system in which we can access and fairly evaluate feed efficiency among our population when they're exposed to these environmental challenges. Through a cooperative agreement, again with the Department of Agriculture, we were able to purchase in 2022 the first Vitelli Sense. It was previously known as the GrowSafe system. And this is at our Upper Piedmont Research Station. And again, it's been in effect since 22, and we've had the opportunity to conduct several types of studies where we're able to put cattle into this system during our summer months or winter months, depending on what our goal is, and then use specific feeding programs in order to expose the animals directly to the toxins that cause phaeococcosis and look at the output. The way this system works is pretty phenomenal. And that is that these animals have an RFID tag placed in their ear, and there are weigh bunks both in the feed bunk system as well as a weigh pan here now in the water monitoring system. So we're able to assess bite by bite or drink by drink how much these animals are taking in. That's then directly communicated to a computer on the site, and that data is then uploaded into the cloud, and we're able to access that data throughout the study. So what we've been focused on then is looking at how these interactions occur on the phenotypes of these animals. And so we're looking in addition to our ergovaline treatment, the weakened effect or the temperature and humidity index, and how does that impact the animal's body weight, average daily gain, and then some phenotypic data here such as hair shedding, pulmonary artery pressure, water intake, feed intake, and so forth. So ultimately, what we're trying to do on the cattle side then is, if we think of this as our focus on residual feed intake, again, draw your attention to the center here where we have our two animals. We have animals, both animals are gaining the same average daily gain. The one here on the left is the superior one, as it is gaining that same amount with less intake. And so what we have then is our dots represent individuals within our population. And so now we're going to want to be able to select these animals that have that low intake with this equal average daily gain and compare those to the opposite side of the animals that have the higher intake with that same daily gain. So these are going to be our superior animals down here in the bottom in that green circle, which we want to continue then to expand that population, understand why they are the superior animals within our system, and then ideally would love to get those out into the population within the North Carolina breeders to expand that group of feed efficient animals and increase the feed efficiency across the whole state. So this is a little bit of what we're doing on the cattle side, and I'm going to turn it over now to Andrew and let him talk about how we've adapted this system to the small ruminant side.
Yeah, so I started at NC State in 2020. I was fortunate and it had been 23 last year to expand the sheep system and kind of complement the beef system in putting sheep flocks at some of our research stations, and we continue to grow those. And along with that, was able to start testing feed efficiency in our sheep populations, similar to what we're doing on the cattle side of things. And so in the sheep industry, we're not a big industry. We don't have the genetic evaluation and the strengths and numbers that we have in some of our other species. If you look at anyone familiar with cattle populations and cattle genetic evaluations, we have an EPD for a large variety of traits. In the sheep industry, that number is minimal. And so the opportunity to start measuring some of these new novel traits in the sheep industry is pretty exciting. I was fortunate when I was in grad school at WVU to utilize the GrowSafe system there, and we had adapted that system to utilize for small ruminants. We tested both sheep and goats during my time there. I was able to take a similar model and adapt our system here at our Upper Piedmont Research Station. And so we've got 8 feeders that have been modified to feed sheep compared to cattle. So they're the traditional cattle bunks. And so we put an insert in there. We put a feeder that holds 50 to 100 pounds of a pelleted complete feed. that sits up where the animal can get to it. And then we build a kind of a stair on the inside of the pen where those animals can get up, walk up, and be able to eat out of that feeder. And the transition between the cattle feeder and the sheep system is really in an afternoon, you can transition the whole system between one species and another. So was excited to have the opportunity to do this and be able to kind of squeeze some sheep projects in between cattle projects, because ultimately the system was funded by cattle industry money, and so I was thankful as the small ruminant person to be able to utilize the system, and we've continued to use it since. So oh, wrong direction. There we go. And so building on this, we're fortunate here at NC State to have the system and be able to use it for small ruminants. And then where we connect into kind of this greater system, I was fortunate to go to grad school at Virginia Tech in West Virginia. And both of those institutions now have GrowSafe systems or Vitelli Sense systems. And so we have the same system at 3 different institutions across actually 4 different research stations. So Upper Piedmont Research Station there in Reedsville, kind of upper, north central North Carolina. We've got 8 feeders there. We've got 8 feeders that are currently being installed at the Upper Mountain Research Station in Laurel Springs. So a higher elevation. We have 8 feeders that went in last year at the research station in Southwest Virginia. They're in Glade Spring, close to the Tennessee line. And then we have the 8 feeders, the 8 original feeders that are there in Morgantown at the Animal Science Farm. And so really across the entire system, and the bottom of the slide got cut off here. But we've got 32 systems across the entire the sheep alliance, I guess you could call it, and the opportunity to feed. We generally think somewhere between 15 to 20 head per feeder. So total one-time capacity is actually pretty good. And the biggest problem we have is we don't have enough small ruminants to keep the system full to its full capacity. And so we're working on that. Wanted to share a little bit of what we've done so far. So this is data on 4 contemporary groups, 229 head that's been tested since the summer of 2022. We tested for about 45 days post-weaning, so sometime between 3 and 5 months of age. I will admit that we do not have enough phenotypes. 229 animals on a trait like residual feed intake, that's an extremely messy trait with lots of variability, is not enough. So we're trying to build numbers there, but it's a start. We got to start somewhere. And so I've grouped these by sire group, and so these are progeny out of one of those sires. This is just representing sires that have 10 or greater progeny that have been tested, and looking at the percentage of those progeny that rank in that low RFI category. So you remember Dr. Poole mentioned lower numbers are better. We want negative numbers for these feed efficiency traits because that means they ate less compared to what we would expect them to. And so those sires that have fewer than 50% of their progeny ranking low RFI, those are the ones we obviously don't like too much. From a research perspective, they're interesting, but from a commercial production standpoint, they're not what we want. We would prefer the animal over here on the right, sire 4, who over the 4 contemporary groups, and he's actually got over 50 progeny now. Over 60% of his progeny will rank in that low RFI category. I was also interested on the female side. We know that mitochondria are the powerhouse of the cell. We know that mitochondria are passed through maternal lines. And so went through and ranked U families and categorized where animals fit into those U families. And just here summarize those U families that have greater than 5 progeny records. Same deal here. We have some U families here on this end where north of 60% of their progeny are ranked in the low RFI category. That U family there on the far right, 100% of her progeny, descendants, I should say, progeny and those granddaughters, grandsons have ranked in that low RFI category. So continuing to explore this, there's still a lot of work to do, but it's a start. So lots of people involved in this, and Dr. Poole already mentioned some of them. It wouldn't be possible without the Department of Ag and their investment in the system, and the research stations and the research station staff, Cattlemen's Association and Ag Foundation, and I've been really thankful as the small ruminant person who doesn't have a large industry backing to be able to work with the cattle folks to be able to do a lot of this work. So
through the advancements that our team has made both on the beef side and on the sheep side, our opportunities are growing. This coming year, we're going to add another 32 feed bunks to these original 8, and we'll add another will total 20 water bunks, again, across 4 different research stations, which gives us the power then to add a lot larger animal groups, continue to keep these full throughout the year, not only collecting the feed efficiency side, which is extremely important, but now with the advancement of the water efficiency, we have the opportunity to be the leader in that because that is a new technology that's coming about. And we're one of the few states that have that investment in that type of equipment. Where we see this going forward, again, is looking at our greenhouse gas emissions. There are adaptations with the system that are still in development to add methane collectors into our bunks. So not in addition to gaining the access to what are they eating on a bite-by-bite basis, when they have their head in the bunk eating, we'll be able to estimate the methane that's being released during that process as well. That will continue to expand our research opportunities. So we appreciate your attention and thank you for your time.
If we could ask the speakers to come down to the front row and take a seat so that we can share the mic. Once everyone gets situated, we can take some questions from the audience.
Hi, question for Chris, Chris Malteca. Hey, Chris. I was wondering, do you think that you could at some point apply the same principles of genomic selection that you apply in animals to microbia or consortium of microbia, or are you thinking about that?
Yes. So, yeah, I haven't presented any of that. There are clearly a few caveats on establishing causality and mediated effects and who's driving what. But yes, and I can give you a little bit of literature if you're interested in how you can do that. Yes.
I'm curious about the peanut flavor project because while statistically you can see differences in peanut flavor trending down, like as in regards to the UPPT test, are consumers actually noticing that trend as well? Because while it may be reflected in the data, is that actually a reasonable, actionable taste that people are noticing?
That is a great question. That is a question that we've asked that we've gotten some very bland answers from most of those people because they don't want to tell us whether or not like their products are seeing like, dips because of flavor or some impact due to it. But I would say that the fact that they came to us asking that question meant that there was probably some consumer perception of flavor that is declining over time. And that's kind of our best guess. Yeah.
I have a question for Emily. So could you expand on how you are phenotyping your gene candidates in rice?
Yeah, so in the Lawrence lab at Arkansas State, they're leading that aspect of the project, but they've got a LabNetTech scanner. So they are phenotyping plants at early vegetative stages with the drought stress experiment, and then they're also growing them up to maturity and phenotyping the seeds after the project. They haven't gotten to that part yet, but yeah, so we'll be looking at mostly at chalkiness and other metrics for the seeds.
And to follow up on that, in terms of your collaboration with universities for the development or phenotyping and genotyping progress, how do you handle the IP or the ownership when there's a collaboration like that?
Yeah, so that's a great question, and I was really interested in the talk before that, from my perspective, I think we've been really sensitive to that as well. Students and professors really depend on those publications, so that to handle that, we just try to set the terms clearly in the beginning of the project, so we kind of in that project, we always knew that our Helios lab was going to be doing the phenotyping and the student would need to have the publication at the end. So with the genomic part, which we handled at Avalo, we carried out our kind of proprietary genetic method. And we also carried out a traditional method that they can publish on as well. So we kind of tried to support both objectives from both sides and balance those in the IP questions. And then we have, NDAs and statement of work that delineate all the kind of outcomes from the research project as well. And for us too, it's been really nice because we're able to investigate some avenues that we don't really have bandwidth to investigate ourselves. So it's kind of a nice way to look at those avenues as well at the same time as kind of accomplishing our business goals. So I think it's a win-win.
Hi, my question is for Drs. Weaver and Poole. About the residual feed intake, is there a plan for you all to look at the microbial side of things, kind of like what Dr. Malteca has done in terms of characterizing maybe the microbiome, looking at different, microbial taxa, etc.?
So great question. One of the challenges we have with ruminants is that the fecal microbiome doesn't necessarily follow the same track as the ruminant microbiome. So requires a little bit more invasive testing in order to do that. But that is something that we have discussed. We're still in the early stages of identifying this population of feed-efficient animals, and that will most likely be the next step since we have a good collaborator in the department.
But actually, now you can use buccal swab to do that, so that might be an option.
Great question.
If there are no more questions, let's thank our presenters. Don't go anywhere.
Given the time, I'm going to keep you just a little bit over. You know how I hate to do that. But it's going to be totally worth it because what we do is we're going to wrap up with 4 2-minute pitch presentations from grad students and postdocs. So they're allowed 1 slide and 2 minutes to present. It's going to go rapid fire. There will be no time for questions, but we have an amazing reception afterwards. All right. So our first presenter is JJ Wang talking about a genome to phenome mixed model incorporating functional annotations. You have 2 minutes. Here it is.
Good afternoon, everyone. My name is Jun Jian. I'm a PhD candidate in Animal Science Department. Today, I'm going to talk about our recent research that a genome to phenome mixed model incorporating functional annotations. Functional annotations are like labels on SNPs. They tell us what regions of DNA are doing, like they are involved in gene regulation or coding for proteins. By looking at these labels, we can know which element of genome that a SNP involved in, with that advancements of functional genomics. We now can access increasingly comprehensive annotation datasets. But how can we do to use these datasets to better interpret genomic architecture behind the phenotypes? In this study, we developed a method that can incorporate functional annotations into a linear mixed model framework, and it's implemented in MPH software. If you'd like more details about this method, please scan the QR code in the corner. A key innovation of this method is that we treat large effect QTL as fixed effects and small effect SNPs as random effects. We use functional annotations to study small effect SNPs. To show how it works, let's take milk yield, for example. With this method, we can calculate the per SNP heritability enrichment, which means that how much a SNP can contribute to milk yield across different annotations. For example, for milk yield, coding sequence region and the conserver region have the highest enrichment. Overall, our method can provide insights into the functional relevance of genomic elements. Meanwhile, as more annotation database become available, our method will become more valuable for studying relationship between genome and phenome. Finally, we'd like to thank USDA EFRI for supporting the method development, CDCP for data, and the USDA ARS for computing resources. Thank you.
Our next presenter is Kairi Tanaka, speaking about harnessing genomes to phenomes for sustainable dairy production. Kairi, you have 2 minutes.
Thank you. Thank you for the invitation. I'd like to share my thoughts about a knowledge gap today with you all. So here's a simple image of a cow grazing on a pasture. But if you zoom into what they are eating, plants are complex both in structure and metabolites. And those plant metabolites are bioactive molecules that microbes see in their gut. And so animals, their evolutionary requirement for animals is to break down cell walls to sort of access the nutrients within. And so the gut microbes are the only ones that can help break down the cell walls. As you can see here, a picture of cellulosome. It's sort of this protein machinery that the microbes employ to break down cell walls. And after that, cascading effect of sort of microbial metabolites come down to some major metabolites such as volatile fatty acids, and even methane is part of that. That influences the host physiology, and in the organs, not only the gut, but also liver and other parts of the body. The cells react by altering their genetic response. So the question is, do we really know what those plant and microbial metabolites are? Not really, or very little. But at least in case of microbes, they know how to deal with those metabolites. So I think we should harness the genome to phenome initiative to figure out what exactly is responsible for the desirable phenotypes in animals by exploring both the plant and the microbial metabolites. Thank you.
Our next presenter is Allison Barnes, talking about advancing maize genomics through lipidomics. It looks.
I think I just accidentally left an illustrator. Yeah, that's fine.
Okay, we'll make it work. Okay, thank you.
Okay. Hi, everyone. I'm Allison. I'm a postdoc with the USDA ARS in Raleigh. And so the project that I'm working on comes out of this larger project called CIRCA, which is the Circular Economy that reimagines corn agriculture. And the idea here is to create corn that's more nitrogen use efficient. One of the ways that we can do this is by planting our corn earlier so that there is less open ground and less loss of nitrogen that way. But in order to plant corn earlier, even if it's just by a few weeks or a few days, we have to increase the cold tolerance of corn so that late season freezes will not impact the yield later on. One of the ways that plants adapt to cold temperatures is through changing their membrane structures. And so I promise you don't need to know anything about biochemistry to understand this. If you just think about different lipids as different shapes. And so there are some that are triangles and some that are rectangles. And the overall goal of the plant is to balance those triangles and rectangles during a time of stress so that there's enough flexibility for the membrane to stay alive and not so much rigidity that the membrane shatters. And so what I'm trying to convince you of is that we can use these membrane lipids as phenotypes in the same way that we would use plant height, ear height, or yield, and then use them in modeling and other breeding programs. And so the way that we can do that is we can scale up from sampling a single plant to looking at a whole host of plots in a field. So in 2023, we did this with 1,500 plots in Clayton, and then we extract the lipids and run them on an HPLC mass spec. And we essentially then get an output of a whole bunch of numbers that can be treated and added to models just like any other trait would be. And so the population that I'm working on now that I think would be of interest to many groups of people is teosinte, cross to B73. Those F1s are then back crossed and selfed. And then we have this great population that has snippets of teosinte genome with a lot of B73, so we can use it for tracking different traits. So thank you.
Final 2-minute picture is near one, our final Tandakar, and there it is.
Good afternoon, everyone. I am Nirwan from the Raisana Alvarez lab, and I'm also a part of the SARCA project, which you guys just heard. And I'm working on something different, but also in the nitrogen use efficiency. So what you see here in the figure to your left is the basic nitrogen cycle. So what happens is we add fertilizers and the plants get their nitrogen from the fertilizers. But what also happens is, like, when you apply a lot of fertilizers, the nitrogen gets changed into nitrous oxide, and they might get runoff to the water streams, and they might also go into the environment as nitrous oxide, causing climate change. Causing high increases of temperature, causing climate change, which is very important, at least depending on what happened last night. But so what we want to do here is we want to be able to utilize the nitrogen more efficiently. And what I mean by that is we want to remove the nitrogen in the seeds and put them back into the leaves and the stalks. And if you remember Dr. Malteka's slide, you see 3 little pigs eating corn there. So we don't want nitrogen in the corn. We want them back into the seeds. So when we ground them back into the soil, the nitrogen can go back into the soil, and we can reuse the nitrogen that we used from the fertilizers. So the way we did that was we took land races that are adapted to the Americas, from South America and North America. And then we have our soil nitrogen maps. And then we ran 2 different types of GWAS, and then we got several different candidate genes. And 2 of the genes that I wanted to talk about is the TCP9 gene and the nitrate regulatory gene. So the TCP9 gene in rice has been identified as an important gene for nitrogen use efficiency, and in modern rice it has actually been removed. So what we want to do is we want to safely utilize or put in some of these genes that we found into the modern maize, and hopefully they will be able to solve the NUE problem. Thank you.
So let's give our 2-minute pitchers another round of applause. Thank you, guys. So I think the Grace Under Fire Award for AV issues goes to Acer and Emily. But thanks everybody for your patience on this. I realize I did not introduce myself. I know so many of you are in the room. My name is Deborah Thompson. My role for the College of Ag and Life Sciences is Research Partnership Development. I love that all of our presenters today showcased their collaborations with an industry partner and how it benefited not only them, but also the industry partner. So thank you all. I appreciate that a lot. Where'd Kathleen go? There's Kathleen. So I partner very closely with Kathleen Denya, who does a similar role with other things for the Plant Sciences Initiative. Feel free to talk to either one of us during the break. I'm going to close the program, but I do need to thank a few people, our presenters and our 2-minute pitchers, of course, NC Biotech for hosting, and Sean for AV support, which I clearly needed today. Lauren, thank you so much for moderating the presentation. Benjamin, you were amazing. I appreciate all of your help up front and in here. And Allison, where did Allison go? She's up front. Allison, who worked with me, she was the subject matter expert, helped me pull together the program and find the right presenters for this. One last thing before I let you go. With the launch of North Carolina State's Genome Editing Center, which you should be hearing about if you haven't already, our showcase in the spring will focus on gene editing. We don't have a date yet. It'll probably be in April of next year. So we look forward to seeing you then. I think I can close the program, and in theory, there's food and drink outside. Thank you so much.